So welcome to this new ecotoxicology webinar. So today we are pleased to welcome Dr. Muhamad Arslan, who currently occupies a postdoctoral position in the Department of Civil and Environmental Engineering at the University of Alberta in Canada. And Muhammad will present his PhD results. The results he obtained during his PhD at the Helmholtz Center for Environmental Research, UFZ, in Germany. So he will talk about antimicrobials in wetlands. So have a good webinar.
Hello everyone, my name is Muhamad Arslan, and as Dr. Stefan mentioned, that I did this work in during my PhD at Helmholtz Center for Environmental Research, and the topic of my research was antimicrobial in constructed wetlands can cause in planta dysbiosis. So this is a very brief introduction about me. I did my doctoral research at UFZ as a guest scientist, and I was awarded with the title Doctor of Natural Sciences by RWTH Aachen University in Aachen. Previously, I did my bachelor in environmental sciences from University of the Punjab, Pakistan. Then I did my master's in biotechnology from Pakistan's National Institute for Biotechnology and Genetic Engineering. Then I did my another master from King Fahd University of Petroleum and Minerals in Saudi Arabia. And PhD, as I mentioned, I did in Germany. Regarding my job activities, I have worked as a teaching assistant at King Fahd University in Life Sciences Department. I've been guest researcher at UFZ, and nowadays I'm a postdoc at University of Alberta. The topic of my research interest are microbial ecotoxicology, host microbe interactions, pollutant transformation in the wetlands, and genomics and transcriptomics of plants and microbial communities. Additionally, I'm serving as a guest subject editor in Advances in Life Sciences Journal and guest editor in sustainability. So far, as of today, I have 461 citations, and this is about me. So here is the doctoral research timeline, how I developed the idea with the help of my supervisor, and how I finished during the 3.5 years starting in 2015. The work was initially it was initiated by a master degree student who was looking into the transformation of two antimicrobial wetlands, and based on his results we developed a hypothesis, and I started doing the first study looking at the response of microbial community inside plant. That study led to further questions which to address them I conducted another study, and there was some delays regarding community analysis, and to support this study I did a third study on transcriptomics and metatranscriptomics. Finally, I completed my PhD in 2019 and defended in June 27. So background, we know that this is the age of micropollutants, and antimicrobials are among huge concern of total micropollutant loads because of their active nature, chemically active and inert nature. We know that they come to the environment from multiple sources, for example, household waste, hospital waste, agricultural runoff, industrial effluents, and all of them, approximately 98%, they come into the wastewater treatment plants without being degraded. And wastewater treatment plants don't have the facilities to remove these contaminants at at a minute level, which is nanogram to microgram per liter. So they come into the environment, exposing wetlands as tertiary treatment system or standalone system. So regarding the selection of antimicrobial, I worked with trimethoprim, which is the combination of two antimicrobials, sulfamethaxazole and trimethoprim. They make 10% of the total antibiotic load in the European wastewater environment. Here you can see a modeled diagram or the map of distribution of these two antimicrobial in European Union, and you can see here that sulfamethaxazole is actually very high or abundant in Germany, lesser in France, and same is the case here, but in the eastern part their consumption is more. So we know that at wastewater treatment plants, they are not primarily removed because of their lower concentration. And after the wastewater treatment plant, we have constructed wetlands often used as a tertiary treatment system. So if the wastewater after the treatment has this concentration of antimicrobial, they may cause or affect the performance of wetlands, hence reducing the efficiency of the system. During this work, I was also involved at a field scale floating treatment wetland solution in Pakistan, in which we developed a floating wetlands covering 20,000 square feet of total area, and its results are published in Nature Sustainability in 2019. So the question here again is, these wetlands which we are using for the treatment of sewage and industrial wastewater for the Faisalabad city in Pakistan, they're receiving antimicrobials as well in the system. So whether the presence of this antimicrobial can affect the performance of these systems, which we are still ignorant as of today. Background. This is the schematic representation how a wetland system works. We know that in wetland we have plants and microbes. Those microbes which are living close to the roots are known as rhizospheric bacteria or rhizospheric communities, microbial community, and those bacteria that lives inside the plant are known as endophytic bacteria. And they work together, both plant microbes work together to reduce the toxicity due to contaminants present in the wastewater by degrading them or transforming them into simple inorganic compounds such as carbon dioxide and water. And they also help plant to avoid any pathogen invasion. They also help plant by producing phytohormones or plant growth promoting activities, and they also help plant by doing nutrient assimilation activities. So microbes have a very important role in constructed wetlands, and we know that if antimicrobials are present in the wastewater, they will selectively target microbes and can affect the system performance, affecting the host health as well, the health of plant. As of before I started this research, there was no information available on response of endophytic communities to antimicrobials. So our hypothesis was based on animal gut analogy, where we know that when humans or animals take antibiotics, the microflora in the gut is disturbed. Initially, after the exposure of antimicrobial, it is wiped out, and after some time, new microflora starts developing, and possibly that is not the that new microbiome is not doing the same function, but what was the initial microbiome doing there. So there is a microbial imbalance, and also there are chances that pathological or pathobionts or non-beneficial microbes or opportunist microbes start developing communities in the gut. This affects the host health, and it has been well investigated in animal kingdom that it leads to dysbiosis, which is a microbial imbalance. And once it is there, it is very difficult to restore the structure and function of original microflora. So the hypothesis was, when antimicrobials are exposed to the plants, and their repeated exposure in the nature can disturb plant microbe interplay or plant endophyte interactions. And the first assumption was that it will inhibit the good guys or good bacteria, which are known as endophytes. They will inhibit their function as well as their abundance, leading to the reduced performance of the wetlands ecosystem. For these two antimicrobials, which are sulfamethaxazole and trimethoprim, they act sequentially and block the production of folate, which are building blocks of amino acid. It is necessary or required for the production of folate or folate metabolism. And once we have this sulfamethaxazole, it competes with para-aminobenzoic acid due to the structure similarity and stop the formation of dihydroteric acid. Then the mode of action of trimethoprim is on the on the last stage, where if some of the dihydrofolic acid is already formed, it competes with dihydrofolate reductase and stop the formation of tetrahydrofolic acid. Together, they are bactericidal, and they stop the formation of folate, which is required for the production of amino acid as well as for the DNA replication. So once they are in the system, there are no DNA replication, no bacteria. The bacteria will not be able to grow or proliferate in the system, and it will cause or lead to the reduced abundance and diversity. So as I mentioned, this study was planned based on some observations made by a master degree student. He was Marcello Santoni from Italy, and doing master thesis research in USAT. He was looking for the transformation of these two compounds in constructed wetlands. And instead he found the transformation. He noticed that the plant health was being compromised. After the exposure of antimicrobial at exposure of sulfamethaxazole at 10 microgram per liter concentration and 100 microgram per liter concentration of trimethoprim, he noticed that the evapotranspiration was decreasing abruptly. And after some pulses at below 1 microgram per liter of the concentration, the plant health dropped further because the evapotranspiration was significantly reduced. The number of green shoots were reducing as well, and there was an insect attack on the plant. So overall, the fitness of Juncus effusus was declined after several exposures. So here two questions were raised. How did co-trimoxazole exposure affect the endophytic bacterial community in Juncus effusus? The second question was, did the exposure eliminate beneficial endophytic bacteria? We know that endophytes in literature are recognized as beneficial guys because they help plant to gain biomass and degrade pollutants and avoid pathogen infection. So if they are eliminated or they are inhibited, the community inside or the host of the health of the host will not be good or sufficient to cope the external stressors such as antimicrobials. And to address these questions, I started my work with cultivation dependent approaches, and then I continued using cultivation independent methods. For the cultivable community comparison, when I compared the microbial community in the unexposed roots with the exposed roots, I found that the community after the exposure was increased both in terms of abundance and diversity. There were some bacteria like Bacillus pumilus. They were not much abundant initially, but they started becoming more abundant after the exposure. Initially, they were the abundance of gamma proteobacteria was very little, but afterwards they were increasing in number. Same was the case with beta proteobacteria, which were not there initially, and they started developing colonies inside the plant root. Similar results were seen for the shoots, but less significant here. The overall abundance was increased and diversity as well, and we found there was a suspected pathogen Pentoa ravandensis, which is previously detected from eucalyptus, causing bacterial blight disease, leaf disease. Here is the comparison of CFU for the roots and shoots. For the roots, the community was almost 10 times higher after the exposure. In the shoot, it was less significant. To ensure that this increase in proportion is not due to cultivation bias, I did qPCR and looked at the abundance in terms of copy numbers of 16S amplicon. And I found that in the unexposed plant, the abundance was lower. And after the exposure in the roots, it was 8-fold higher than the unexposed. I also did some comparison with the natural community from the plants which are grown in the natural environment to ensure that the bias. This is not due to the seasonal bias or due to natural variations as well, and the results were consistent to the systems, the unexposed plants in the controlled reactors. The results were significant for the roots, but not for the shoots here. For the copy number, gene copy number. Again, gamma proteobacteria was found highest, followed by firmicutes and acidobacteria. So here was the next question was new community really beneficial for the host, or it was comprised of opportunistic or pathogenic bacterial species? To address this question, I looked at at different plant growth promoting activities of the bacteria. For example, production of indole acid, phosphorus solubilization, siderophore production, and ACC deaminase activity, and these all four activities are typically used to look how the microbiome is beneficial for the plant, because these are the they produce phytohormone which help plant to gain biomass. And I found that most of the bacteria were able to do ACC deaminase activity, then phosphorus solubilization, indole acid production, and siderophore production. But these results were conducted in vitro, and it was not a complete reflection or true reflection that it really happened in situ as well. And to contradict this hypothesis, I looked at the production of reactive oxygen species in the roots, and I found that there was a high intense signal for ROS, reactive oxygen species, and reactive nitrogen species. And it was in the same region where the microbes were making colonies. So in the unexposed root, there was no ROS production. In the exposed root, there was ROS production. So from here, it became clear that plant was not liking this community, and it was resisting or defending against them. And when we did this, when I did this ROS study, there were no antimicrobial in the system. It was done after the system was exposed, after 3 months or more. So the primary assumption was there were still microbes which plants were not liking. So there was, in the end, host defense system was activated after the infection, invasion of new species. Then the next question was, how were endophytic bacterial communities spatially localized before and after the exposure? Where they are typically found. And I found that in the unexposed plants, the vascular bundles or xylem tubes, they were empty. But after the exposure, these new microbes were making biofilms. You can see here, this is a three-dimensional representation. And the most abundant group was again gamma proteobacteria, followed by firmicutes. Then the next question was, how stable was the endophytic communities before and after the trimethoprim exposure? And to address this question, we did 16 assembly con sequencing at genus level to dig further into the microbial physiology information and diversity composition and statistical interpretation. For this part, I conducted Fisher alpha diversity. I computed Fisher alpha diversity index and found that in the root, unexposed and exposed root, the diversity didn't change much for the roots, but for the shoots it reduced significantly. When I did NMDS plotting, non-metric multidimensional scaling plotting, and I found that before the exposure, microbial community in the unexposed root and shoot was clustered together. But after the exposure, it was having two different clusters for the roots and for the shoots. So from here, it became clear that the community was definitely not similar what was there before. And going back to the first hypothesis or the question that whether the beneficial community was inhibited or good guys were not there after the exposure, I plotted a heat map for the OTU operational taxonomic units, and I found that initially the community which was in the unexposed root was inhibited or disappeared after the exposure, and there was totally new community developing in the system. This was more prominent from the roots and less prominent for the shoots, but still in the shoots, the bacteria which were there were not there after the exposure. So at genus level characterization, three primary observations were made. The first one was there was sulfur oxidizer highly abundant in the system. They were comprising approximately 50% of the total community. Second observation was there was a flux of C1 compounds after the exposure, and it was increasing because methylocyst, which feeds on one carbon methanol or similar compounds, one carbon compounds for the metabolism, they were increasing after the exposure. There was a vigorous iron cycle. Initially, there were iron oxidizers, but after the exposure, iron reducers also started developing communities, and they were making from 2% to 10% of the communities after the exposure. There were several further questions of like how the bacterial community changed temporally, because this was the observation made or compared initially and after the exposure. So what happened temporally? Why we have high sulfur oxidizer abundant? That was the second question. The third question, was there oxidation of plant-derived methanol, or was there any role of rhizospheric methane diffusing into the plant root after the exposure? The fourth question was, was there any role of rhizospheric community in shaping the endophytic community? And to address all these questions, I conducted a detailed, in-depth second study exposing my plants with the 0.1 microgram per liter concentration of sulfamethaxazole and 0.03 microgram per liter of trimethoprim. And then in the next phase, the concentration exposure was increased to 10 microgram of sulfamethaxazole and 3 microgram per liter of trimethoprim, and then finally to 50 microgram of sulfamethaxazole to 17 microgram per liter of trimethoprim, and 100 microgram, 100:33 microgram per liter for sulfamethaxazole and trimethoprim. And what I found in this temporal exposure, that the plant evapotranspiration was not dropped significantly unless the plants were exposed to 50 microgram per liter concentration of sulfamethaxazole and 17 microgram per liter of trimethoprim. For the reactor, for the first reactor and for the second reactor, the observation started appearing a bit earlier. However, in the both reactors, the behavior was similar, that evapotranspiration started decreasing. For the phase 6, there were no antimicrobial in the system, but plant evapotranspiration was again decreasing continuously. I also counted the number of green shoots for the plants in the reactor, and they showed the similar behavior, and looked at the chlorophyll fluorescence signal, and similar observation were made here again. That in the phase 4, when the system were exposed to 50:17 microgram per liter of sulfamethaxazole and trimethoprim, there was a significant effect on plant health. Initially, the roots were very strong and intact and brownish, but after during the phase 4, they started turning porous, dark brownish, necrotic, and finally blackish. And there was an insect attack after the exposure to the plant. Here you can see that the plant's color is very green, and after they started becoming more brownish. To answer the question I mentioned in the earlier slide, I did cultivation independent analysis of 16 amplicon sequencing for each phase. And here I found the temporal response of endophytic community as well as the effective concentration of antimicrobial for the endophytic communities, which can inhibit the microbiome. From the Fisher Alpha Diversity Index, I found that the community initially, the diversity of the bacterial community initially was decreasing and then started increasing. This was seen in both reactors, but one reactor showed significant results. It was the second reactor. So this decrease in alpha diversity was in accordance to what we know from animal kingdom, that when we have a decrease in alpha diversity, it is the cause of dysbiosis, or it leads to dysbiosis. That's a proxy of dysbiosis. And the decrease in diversity was already starting at low concentration, which are like in phase 1, 0.1 to 0.03 for one reactor, 0.1 to ratio 0.03 microgram per liter of sulfamethaxazole and trimethoprim. But the effective concentration were in the phase 4, when both reactors displayed that there was decrease in community, and it was in 50:17 microgram per liter of both antimicrobials. Okay. And to confirm that this initial decrease and then increase was not the restoration of original communities, I did principal component analysis at dose-dependent fashion, and here I found that the phase 1, 2, 3, and control, they are clustered together, but phase 4, 5, they are clustered here, and phase 6A. For the phase 6B, when the evapotranspiration was almost zero, the community was very different, and I consider that this was the post-secondary effects and due to the death of the plants, many plants. Here you can see that the community was not much disturbed, but after the exposure of 10 microgram per liter, the community didn't develop what was there before, and there were opportunists or pathogens taking advantage of the weakened system and feeding the system. I Also looked at rhizospheric bacterial communities to understand what is going on, and I found that many of the rhizospheric bacteria were anaerobes, essentially doing the same function what we have seen in the animal gut. Even some of them, like Litori-Brevibacter or Lactobacillus, they have been previously identified from the animal gut. So what they were doing, they were fermenting the complex organic compounds in the roots, in the form of which are coming in the form of root exudates. That was the picture when the system was not exposed to the antimicrobials. But after the exposure, if you see here, their abundance was decreasing. For some of the anaerobes, it was decreasing. You can see here. So how the overall system was changed, or this is shown a model diagram. This is a plant root, and this is a typical for the Juncus effusus. It's a helophyte, which means it deposits atmospheric oxygen into the rhizosphere, and plant root exudates are also produced in the are also released in the rhizosphere, and with the help of rhizospheric anaerobic bacteria, they are being fermented to methane or hydrogen molecule. There was because I was using artificial wastewater, there were inflow of benzoate and acetate as well, which might be helping the anaerobes as well to proliferate. So there were possibly methane diffusion from the rhizosphere to the endosphere. When the system was happy, there were hydrogen oxidizers in the endosphere. As I mentioned, that one of the question was there were sulfur oxidizer, approximately 50% of the total community. And this question was explained here because these sulfur oxidizer also use hydrogen molecule as an electron donor. So possibly this hydrogen was diffusing into the endosphere on the plant root and helping the endophytes to proliferate. This is the picture for undisturbed system. There are more hydrogen oxidizers and very less anaerobic bacteria or within the root. But after the exposure or in the disturbed root, there were more heterotroph coming up in the system. Aerobes were decreased in the phase 4. For example, if you see here sulfur italia, its abundance was decreased. Methylocyst, its abundance was increasing. So sulfur italia is hydrogen oxidizer. Initially very high, but then decreasing. Methylocyst, initially very low, but increasing. Same is the case with the sulfuric curvum, hydrogen oxidizer. Abundance decreasing. Ironella decreasing. Spirocheta decreasing. Hydrogen phaga decreasing. So in the end, it became clearer that during the phase 4, major changes were seen, and inside the plant root, the conditions started becoming anoxic, or there were some anoxic niches or anoxic patches developing within the roots, which were not favorable for the plant endophytes. Yeah, these observations were made at the genomics level, and to support this study further, I carried out the transcriptomic and metaproteomic study as well to look further that what is the response of plant and microbial community in terms of proteins being produced or present in the system, whether there are some stress related proteins or what is going on. For this work, I had to develop the database of Juncus effusus transcriptomes by doing transcriptome sequencing of different varieties of Juncus effusus across European Union. And this was done with the help of our collaborator at UFZ Halle, Dr. Stefan Michalski, who was doing the SNP analysis for Juncus effusus and he had sampled and sequenced 18 genotypes of Juncus effusus from different part of the European Union. I used the same data set and annotated it using different databases, protein domain matching, PFAM, RFAM, non-coding, RNAs, or the DB database, followed by quality testing via Busco and or other statistics. Here is the complete illustration of what I did, and this part is done by Dr. Stefan Michalski group, and this part is done by me, the lower part. And what we did in the end, we were able to annotate 70% of the contigs or transcripts in the assembly, in the transcriptome assembly. The quality score, which was detected via benchmarking single copy orthologs, Busco, was 81%. And when it was compared with the quality score of well-finished annotated transcriptome of sorghum bichlor and Oryza sativa, we found that our quality score was sufficient. For sorghum, it was 82%, and for Oryza sativa, it was 78%. And there were 7,000 protein sequences identified through CAC analysis. For the annotation part, when it was completed, 111,567 contacts were annotated, and we found that there were 30,932 unique genes among these contacts. I compared computationally or statistically the quality score using David, and I found that there were 3,185 enriched sequence. I also compared the results of orthologs genes using orthoven pipeline, and I found that we had a very much similarities with the phylogenetic closer relative of Juncus effusus, which are sorghum bichlor, Oryza sativa, Z mice, and wheat. Yeah, is the further explanation. So the comparison with the three closer relative of Juncus effusus, which was sorghum bichlor, Oryza sativa, and Z mice. Vigo comparison showed that Juncus effusus had distribution of genetic feature much similar to what is previously reported for Oryza sativa and sorghum bichlor. Once again, suggesting that our annotation was much successful. For the orthoven or cluster of orthologous genes comparison, there were 9,872 unique genes identified for Juncus effusus and 10,296 genes shared by all members. And there were some similarities showing sorghum bichlor highly similar to the Juncus, then Oryza sativa, and then Z mice. I also looked at the completion of plant pathogen defense pathway and found that it was complete for the hypersensitive response, for the programmed cell death, and for the defense related genes, which was the assurity that we can use this database for our stress response study as well. And finally, I compared the proteomics results, which were proteins were extracted from the first experiment, unexposed root and exposed root, unexposed shoot and exposed shoot. And we looked at bacterial, archaeal, and plant proteins. Here, I found that there were two there were if you see here that the bacterial proteins after the exposure increased for the roots, and for the shoots it was less significant. For archaea, they were not there in the unexposed root, but started coming in the exposed root and exposed shoot. For plant protein, the results were not significant because the extraction of protein was less efficient after the exposure from the roots as compared to the shoots. Plant proteins were typically related to normal process of plants such as photosynthesis, glycolysis, citric acid cycle, cell division, etc. But bacterial protein showed that it was in accordance to what we saw at the genomics comparison, that there were proteins from C1 metabolism pathway, as I mentioned, possibly derived from methanol. There was more bacteria after the exposure feeding on the methanol coming from the degradation of hemicellulose or pectin. There were some membrane proteins, dehydrogenases, reductases, and sulfhydrolase and synthetase. Archaeal proteins were not there, but they were present after the exposure. And this was the indication that there was some ammonium oxidizer. We were always curious whether there are methanogens inside the root before or after the exposure, but we didn't detect methanogens with PCR analysis. However, there were some in the rhizosphere. So possibly there was diffusion of methane from the rhizosphere as well as ammonium oxidizer present in the root. Conclusions. Trimethoprim exposure affected the endophytic community in Juncus effusus, particularly in the roots. Endophytes are always not necessarily beneficial. They could be opportunist pathogen. It has been previously stated in the literature that endophytes are always good guys, but recently this definition is changing, and it is being discussed by endophytes people around the world, that they're saying that they're not necessarily beneficial. And we also go support this new definition that they are not always beneficial. They can be opportunistic. And there is an imbalance in plant endophytic community, which is similar to the gut dysbiosis because of reduction in alpha diversity. And of course, this led to the decline in fitness of Juncus effusus. My study has established the database of Juncus effusus, which is available publicly at Mendeley repository and can be used for omics studies for this model wetland plant. And proteomic studies observations verified at genomics level, especially for bacteria. However, to understand better what was the response of plant endophyte interplay, this should be investigated in depth. In the end, I'm always thankful to my very kind and helping supervisor, Dr. Joachim Muller from UFZ, our partner from Helmholtz Center in Braunschweig, Dr. Dietmar Pieper, Dr. Thomas Noe from UFZ Magdeburg for fish analysis, and Dr. Upendra Kumar Devisetti from Arizona University, USA, who helped me with transcriptomics. And my university supervisor from Aachen, Professor Rolf Altenburger, and Professor Henner Hollard, and other team members. Thanks. If you have any questions.